ॐ सहना भवतु सहनौ भुनक्तु सह वीर्यं करवावहै तेजस्विनावधीतमस्तु मा विद्विषावहै ॐ शान्ति शान्ति शान्तिः।
Last class we started about the biological processes for water and wastewater treatment, and we have seen that the treatment processes, the selection of the treatment processes is based upon the nature of the pollutant present in the water, either it is biological or physico-chemical processes. Okay, because when we talk about wastewater treatment, why we are going for biological processes is the majority of the solids are organic in nature. So, it is very easy to remove the organic matter by the help of microorganisms, and we were discussing about what are the different types of reactions, or how can we classify the reactions based upon the kinetics. So, we have seen what is a zero order reaction, first order reaction and second order reaction, and we have also discussed, or we have also discussed that in biological processes we assume that most of the reactions are of first order, though many times it may be deviating. And we have also discussed what are the different reactor configurations available. So, we discussed what are the advantages of plug flow reactors and CSTR reactors, and what is a CMBR reactor. And it's seen that plug flow reactor needs less volume compared to a CSTR reactor if we want to achieve the same degree of treatment or same degree of removal. Today we will discuss about the biological process in detail, or we will be starting on activated sludge process. So, coming to the secondary treatment, we have already discussed what is a primary treatment. So, the primary treatment is based upon physical unit operations. That means the gravity force is the predominant force there. So, it will be removing the solids which is having high density or high particle size. And we have also discussed about primary sedimentation tank usually employed in wastewater treatment, and it is working on type 2 settling. The design procedure we have already discussed when we were talking about sedimentation tanks. And in wastewater treatment, the primary sedimentation tank remove around 50 to 60% of the suspended solids, but coming to the soluble BOD, the removal is almost negligible. And what we do with this sludge whatever is collected in the primary sedimentation tank, it is of organic nature, or most of them are organic matter. So, we cannot directly dispose them off. So, usually what we do is take this sludge along with the secondary sedimentation tank sludge and send it to sludge digesters, where the sludge treatment or sludge stabilization will be taking place under anaerobic conditions. And as a result we will be getting biogas or carbon dioxide and methane and stabilized sludge. This stabilized sludge can be put into sludge drying beds and afterwards can be used as manure, or you can use it for landfilling also. So, these are the typical values of a primary sedimentation tank which is commonly used in wastewater treatment processes. The detention time varies from 1.5 to 2.5. As we discussed earlier, if the detention time is higher, what will happen? Whatever is the organic matter that is settled in the bottom of the tank starts degrading anaerobically, and that generates gas. That gas will be creating a turbulence in the sedimentation tank, and it will be affecting the efficiency of the tank. And overflow rate varies from 30 to 48, and wear loading is in the range of 125 to 500. and these are the dimensions of the tank. Depth of the tank varies from 3 to 5 meters, length from 15 to 90 and width 3 to 24 meter, and typical value is 6 to 10 meters, and sludge scraper speed is around 0.6 to 1.2 meter per minute. And if it is a circular tank, the depth is the same as that of a rectangular tank, that is 3 to 5 meters, and diameter 3.6 to 60 meter, and typical values varies from 12 to 45 meters, and bottom slope is 60 to 160 mm per meter, and typical value is 80. So, this is the primary sedimentation tank design parameters. It is almost similar to the primary sedimentation tank of water treatment, but here we are assuming it is type 2 settling. So, we cannot use Stokes law for the design of the sedimentation tank of wastewater treatment. And yesterday we talked about many secondary treatments, and we have also seen that in certain secondary treatment units, they do not require a primary treatment at all. For example, oxidation ponds and aerated lagoons, because that system itself will be acting as a primary treatment unit also. Everything will be settling down in the bottom of the tank, and we are giving such a large detention time. So, the particles whatever is coming, either it is suspended or it is inert material, everything will be getting suspended, everything will be getting settled, and the tank itself will be acting as primary and secondary treatment unit. Now, we will see in detail what is this secondary treatment. Each unit we will be seeing in detail. Okay. So, secondary treatment in wastewater, that too in domestic wastewater, mostly biological treatment is being employed. And the reason is the most of the solids are organic in nature. So, biological treatment is the most cost effective one, and the organic matter whatever is present in the wastewater, that is highly degradable organic matter, or we take the BOD COD ratio, the ratio is very high. So, we can go for biological treatment. And when we talk about the biological treatment, we can classify the biological treatment into two different categories. One is suspended growth system and another one is attached growth system. In suspended growth system, entire biomass is in suspension. For example, activated sludge process is a suspended growth system, and other suspended growth systems are aerated lagoons, oxidation ponds, and nitrification and denitrification processes. And coming to attached system, trickling filters is an example, and another one is rotating biological conductors. Anyway, we will be discussing each of this unit in detail, how the design parameters, how to design, what are the important factors we have to consider, etc. we will be seeing in detail. So, before going to the biological treatment systems, now we should know what are the nutrient requirements for the microorganism, what are the environmental conditions conducive for the microorganisms, and how the metabolism of microbes are taking place, and what is the relationship between the microbial growth and substrate utilization, because in any biological treatment process when we talk, the microorganisms are the workers. So, we have to create conducive environment for the workers, otherwise they will not be doing the work properly. So, our treatment system will not be performing better. So, first we will discuss about the nutrients. Okay, we have seen the microbial growth curve and most of the biokinetic parameters in the previous lectures. So, what is the purpose of this nutrient? The nutrient is essential for supplying material for the creation of cell material. That is very important, because from where they will be getting the carbon molecules for the creation of the cell material. So, for that nutrients are required, and the microorganisms require lot of energy. So, this energy source is also from the nutrients. And the last one is how the microorganisms are generating the energy. They are generating the energy by a series of biochemical reactions. Most of them are oxidation reduction reactions. So, if the oxidation reduction reaction has to take place, there should be one electron donor and one electron acceptor. Unless the electron donor and electron acceptor are there, oxidation reduction reactions are not possible. So, in aerobic system, the organic matter will be acting as the electron donor. So, the organic matter will be giving the electrons. So, that will be passing through a series of steps, and finally, the electron should be accepted from the system. So, who will be accepting the electrons? So, in some cases the nutrients itself will be accepting the electrons, or sometimes it will be oxygen in aerobic system. So, these are the essential things required for the microorganisms. Now, coming to the environmental parameters, the pH, temperature, etc. should be conducive. We know that microorganisms are not able to survive either at low pH or at high pH. So, when we were discussing about disinfection, we have seen that if the pH is reduced below 3, or if you increase the pH above 10, most of the microorganism will die. So, when you talk about the biological system, we should see that the microorganisms or the pH conditions in the system are within this limit, and we should be able to provide the optimum condition. Optimum condition is most of the time it will be near the neutral pH. And coming to the temperature, okay, we all know that the enzymes or the proteins, whatever is present in the cell of microorganism, will be getting denatured at high temperature in most of the cases. But there are microorganisms which can survive at very high temperature and very low temperature. But most of the microorganisms, whatever we are employing for this wastewater treatment, they comes under mesophilic temperature range, that or mesophilic microorganism. That means their optimum temperature is around 35 to 37 degrees centigrade. So, if you reduce the temperature or increase the temperature, the microbial activity will be reducing. Okay. It has shown that for every 10 degree change in temperature, the activity will be varying 100%. So, we can see that how the environmental condition is going to affect the microorganisms. Now, coming back to the nutrients itself, okay, depending upon the nutrient selectivity, we can classify the microorganism into various categories. The nutrients will be acting as an energy source, as an electron acceptor, and as a carbon source. So, first we will see the carbon source. Okay. The microorganisms can use either organic matter as the carbon source or inorganic material like carbon dioxide or bicarbonate as the carbon source. The organisms which are using inorganic carbon as their carbon source for cell synthesis are known as autotrophic microorganism. Once again, the microorganism which use inorganic carbon for their cell synthesis, it is known as autotrophic microorganism. The microorganism which use organic matter or which takes carbon from organic matter, those are known as heterotrophic microorganisms. Now, coming to the energy source, they can derive energy from sunlight, they can derive energy from chemical reactions. Okay. The microorganism which derive energy from sunlight is known as phototrophs, and the microorganisms which derive energy from the chemical reactions, that those are known as chemotrophs. And in chemotrophs itself, okay, the microorganism which use organic matter, it is known as chemo-organotrophs or chemoheterotrophs. And the microorganism which require chemical reactions for the energy, but they use inorganic compounds for these reactions, they are known as chemoautotrophs. So, we have seen different types of microorganism. One is autotroph, another one is heterotroph, and depending upon the energy requirement, we can see that they are phototrophs, chemoautotrophs, chemoheterotrophs. Phototrophs are the one which generate the energy from photosynthesis. Just like plants, there are many microorganisms which can get energy from the photosynthesis reactions. And autotrophs are the one which use inorganic carbon for their cell synthesis, and heterotrophs are the one which use organic matter for their cell synthesis. Now, coming to the electron acceptor, okay, we have seen that for any oxidation reduction reaction, there should be one electron donor and one electron acceptor. So, when we talk about wastewater treatment, most of the cases the electron donor is the wastewater itself or the organic matter present in the wastewater. But who is the electron acceptor. Electron acceptor can be either oxygen or inorganic compounds containing oxygen like nitrate, sulfate, etc. So, they can also act as electron acceptors and get reduced. And third one is the organic matter itself can act as electron acceptor. So, depending upon the electron acceptors, we can divide the processes into different categories. Those are aerobic process, anoxic process and anaerobic process. In aerobic process, oxygen, molecular oxygen is the final electron acceptor. So, organic matter releases the electron and it will be traveling through a series of reactions, and because of this oxidation reduction reactions, lot of energy will be released that will be utilized for the metabolic purpose of the cell, and finally the electron will be accepted by molecular oxygen and it will be getting reduced to water. But in certain cases when oxygen is not available, okay, so inorganic compounds like nitrate, sulfate, etc. can accept the electron. For example, if we talk about nitrification, denitrification, what is happening? Nitrate is getting converted to nitrogen. This is very essential when we talk about high ammonia containing wastewater, because there is a limit for nitrate discharge to the receiving waters. So, if you have excess nitrate, how can we remove that one? Okay, we can use biological process for the nitrate removal. That process is known as denitrification. So, denitrification is an example of anoxic process. So, here no molecular oxygen will be available. So, nitrate will be acting as the electron acceptor. Once one compound accepts the electron, what will happen? It will be getting reduced. So, nitrate will be getting reduced to nitrogen gas. So, those processes are coming under anoxic category. Now, we will see what is anaerobic. Anaerobic means either oxygen or inorganic compounds which is having oxygen are not present. So, organic matter themselves will be acting as the electron acceptor. For example, in anaerobic process of wastewater, what will happen? The organic matter itself will be acting as the electron acceptor and get reduced to methane gas. So, that is what we are getting biogas or methane gas from anaerobic processes. So, these are the various classifications. So, these classifications are very important, because when we select the microorganisms for a particular treatment, we should know what type of microorganism we want and what type of microorganisms can use that particular pollutant and convert it into environmental friendly byproduct. So, the important terms one has to remember is what is an autotroph, what is a heterotroph, what is phototrophs, what is chemoautotrophs and chemoheterotrophs, and what is the difference between aerobic process, anoxic process and anaerobic process. Now, we will come back to our activated sludge process. Okay, this is the most commonly used biological process for domestic wastewater treatment. So, here what is happening? The wastewater is there, it is coming, and we are aerating the wastewater. The wastewater will be having lot of microorganisms. So, what will happen? When we supply sufficient quantity of oxygen, the microorganism will be utilizing the organic matter present in the wastewater and they will be converting it into carbon dioxide, water and more and more cells. So, that is what is happening in activated sludge aeration tank. So, in aeration tank, the dissolved form of organic matter is getting converted to colloidal form of organic matter or new cells. So, all these cells are discrete in nature. So, if you let them out like that, there will not be any COD or BOD removal as such, because we are synthesizing more cells into the system. So, the secondary sedimentation tank of the activated sludge process is very important. And if you see the aeration tank of the activated sludge process, what is the nature of the microorganism present in that? Okay, it is a mixed population. So, that mixed population of microorganism will be utilizing the organic matter whatever is present in the wastewater, and more cells are generated and it is coming to the secondary sedimentation tank. Okay, and here we have to separate the cells. So, how can we separate the discrete cells? It will be very difficult. So, it is seen that when the microorganism reach a particular growth stage, okay, we have discussed about the growth curve. Are you remembering? We have seen the lag phase, low growth phase, stationary phase and endogenous or declining growth phase. So, it was found that if the microorganisms reaches the end of that stationary phase or start coming to the endogenous phase, they start producing some extracellular polymers. These extracellular polymers are having some binding property. So, this binding, because of this binding property, what will happen? All these colloidal cells will aggregate and form flocs. So, once they are in, they form the flocs, the settling will be very easy. So, in the sedimentation tank what will happen? The flocs will be settling and we get clear effluent and that can be removed. And this sludge also we have to treat it carefully because this sludge is containing lot of active biomass, so we cannot just discharge it. So, some sludge will be recycled to the aeration tank and remaining will be sent it to the sludge digesters. Okay. And what is the sludge and why it is called as activated sludge process? This sludge is nothing but microorganisms having high activity. Since the sludge is having activity, that is why it is known as activated sludge. Okay. And this is the reaction taking place in the aeration tank. Organic matter plus active sludge or microorganism plus dissolved oxygen gives stable compounds acceptable to biosphere plus increased concentration of biomass. The stable compounds can be either carbon dioxide, water, nitrate, phosphate depending upon the compound or depending upon the nature of the wastewater. So, what is happening in the aeration tank? Soluble organic matter coming to the secondary treatment unit is converted to settleable organic matter. Okay. So, this is what is happening in aeration tank. So, if you take the aeration tank alone, okay, we cannot call it as a treatment system because what is happening, only the nature of the solid is getting converted from the soluble form to colloidal form. So, if you want to get the removal of the solids, okay, we have to have the sedimentation tank. Okay. So, sedimentation tank or secondary sedimentation tank is an integral part of activated sludge process. And we have already seen that the settling tank or the secondary sedimentation tank has to be designed based upon sound settling because the particles are flocculent in nature and it is concentrated suspension. So, concentrated suspension with flocculent particle, okay, it is coming under zone settling, and towards the bottom of the sedimentation tank the concentration of the particles are so high. So, there we will be experiencing the compression settling. Now, we will talk about the ecology of the activated sludge process. We all know that it is not a pure culture system. Not only one single type of microorganism is present. There are multiple groups of microorganisms present which are responsible for the conversion of organic matter into carbon dioxide and water and more cells. And if you see the ecology of activated sludge process, we can see that mostly it is bacteria which is converting the organic matter into carbon dioxide and water, but there are protozoa as rotifiers and fungi present in the activated sludge process. So, if you see, it is a heterogeneous microbial mixture, mainly Pseudomonas and Zuglia are present. Okay. Both these microorganisms are heterotrophic in nature. Heterotrophic means they use organic matter for their cell synthesis and the energy is derived by oxidation reduction reaction, and here also organic matter is being used. But in activated sludge process also there can be autotrophs, for example Nitrosomonas and Nitrobacter. These microorganisms are coming into picture whenever we talk about ammonia oxidation, because we know that protein contains lot of amino acids, and so naturally the wastewater will be containing lot of organic nitrogen. So, if you aerate the wastewater for long time, what will happen? First, all the organic matter that will be getting oxidized, and afterwards this ammonia, organic nitrogen, whatever is present in the wastewater will be getting converted to ammonia, and that ammonia will be further oxidized to nitrite and nitrate. So, this nitrosomonas and nitrobacter are responsible for the oxidation of ammonia to nitrite and nitrate. So, these are autotrophs, that means they use only inorganic carbon for their cell synthesis, and their energies and energy is also generated from chemical reactions, but inorganic substances. And coming to this protozoa, rotifers and fungi, we have seen that mainly microorganisms or bacteria are responsible for the removal of organic matter whatever is present in the activated sludge, present in the wastewater. But what is the role of this protozoa, rotifers and fungi? Okay. Protozoas are very important. They are known as polishers or scavengers of activated sludge process. What they do is, this protozoa are predators of bacteria. So, what will happen? The activated sludge process, we have seen that the floc formation is there. So, once the floc is formed, it is very easy to settle the sludge, but there will be some bacteria which are present in the liquid as discrete particles. So, if the discrete particles are coming to the sedimentation tank, it will not be settling down. So, what will happen? Those bacteria will be coming out along with the effluent. So, naturally, when there is the solid present in the effluent, that too organic, the BOD and COD of the effluent will be very high. So, naturally, you will not be getting that efficiency of whatever you require to meet the effluent discharge standard. So, what this protozoa does is, they will be eating away the discrete microorganisms or discrete bacteria whatever is present in the system. So, what happens? In a way, it is polishing the effluent what is coming out of the secondary sedimentation tank. That is why those are known as polishers or scavengers. So, protozoal presence is also an advantageous thing for activated sludge process to increase the efficiency. Now, we will see how the microorganisms are utilizing the organic matter present in the system, okay? Because we have seen that we are aerating the system and lot of organic matter either in the colloidal form or dissolved form is present in the wastewater, and we have seen that the bacteria are using the microorganism, bacteria are using the organic matter. So, how they are using the organic matter? What are the steps involved in this one? So, it is basically a 4-step process. The first one is adsorption of soluble and colloidal substrate on floc surface. So, whatever is present in the system, it has to get adsorbed to the microorganism. Then after the adsorption, what will happen? The organic material will be getting transported from the from outside the cell to inside the cell. Okay, cell wall, it has to pass through the cell wall and the cell membrane. Okay, because if metabolism has to take place, it has to reach the interior of the cell. So, once it reaches, the transport takes place, then metabolism will be taking place. Okay, so that is the third process, and the fourth one is solid liquid separation. But we have seen that the wastewater will be containing dissolved solids as well as colloidal solids. But if the colloidal solids get attached to the cell, okay, it will be bigger in size. So, how can they penetrate into the cell wall? It will be very difficult for them to penetrate into the cell wall. So, how the cell is able to handle these colloidal particles? It is very interesting. What they do is, the cell will be creating some sort of an enzyme, okay, that will be hydrolyzing the organic matter which is in colloidal form. So, once it hydrolyzes, the hydrolysis is nothing but adding water molecule to the organic compound, and because of that one, it will be splitting into small parts. So, once the hydrolysis is done, so the molecules will be smaller in size, so they can easily penetrate to the cell. So, once it penetrates into the cell, metabolism will be taking place depending upon the condition, either it is aerobic, anaerobic or anoxic process. Then because of the metabolism, energy will be generated and more cell will be synthesized. So, in your system, the organic matter whatever is present in the dissolved form or colloidal form will be getting converted to new cells. So, the last step is solid liquid separation, because the cells as such, if you allow them to go out of the system, the treatment will not be proper. So these are the 4 steps what we usually takes place: adsorption of soluble and colloidal substrate on floc surface, transport of substrate across the cell membrane, then metabolism in the cell, then last one is solid liquid separation process. Okay. And we have already discussed about the agglomeration of bacteria. Agglomeration of bacteria is possible because of biopolymers. So this biopolymer secretion is depending upon the nature of the system. Okay. If all the microorganisms are in the low growth phase, they will not be generating any of these biopolymers. So it is very, very difficult to separate the microorganism from the liquid. But if they are in stationary phase, the end of the stationary phase or endogenous phase, this biopolymer secretion will be very high. So it is easy to separate the microorganism as flocculant sludge. So this is a typical activated sludge process. You can see the wastewater is coming here, and this is the aeration tank. Here what is happening? We are giving lot of oxygen. So under aerobic conditions, the organic matter is getting converted to carbon dioxide, water and new cells. And from the aeration tank, entire liquid is going to the secondary sedimentation tank. So here settling will be taking place because lot of extra sludge will be generated, and from here we will be getting clear effluent, and a portion of the sludge will be wasted, that will be going to anaerobic sludge digester, and a portion will be recycled to the aeration tank to maintain a constant biomass concentration in the system, because we have to provide more and more workers, then only the process will be efficient. So the purpose of recycling activated sludge process is to increase the biomass concentration in the aeration tank. Now we will see how can we design a activated sludge process. You know what is the amount of wastewater coming to the system and what is the amount of BOD, ultimate BOD of that wastewater, because whenever we talk about biological wastewater treatment processes, we are interested only in the BOD, because BOD gives us the amount of organic matter that is biodegradable. But if you talk about COD, that will be giving you all the compounds which is present in the system, either biodegradable or non-biodegradable. So if you want to decide whether we have to go for biological processes or physico-chemical processes, what we have to do first is find out the BOD of the system, BOD of the wastewater and COD of the wastewater, and see what is the BOD by COD ratio. If BOD by COD ratio is more than 0.5, we can go for biological processes. That means whatever is the total organic matter present in that system, above 50% is biodegradable. So naturally we can go for a biological system and followed by some other treatment. When we talk about the domestic wastewater, this BOD by COD ratio varies from 0.6 to 0.8 depending upon the wastewater. That means it is highly biodegradable. So we will discuss about the design of activated sludge process based upon kinetic principles. We have seen that what are the biokinetic parameters of microorganisms. So based upon that one, we can design the activated sludge process. So if you want to make a mass balance of liquid, solids and biomass, okay, we can write something like this. This is Q, this is the quantity of wastewater coming into the system, and S0 is the substrate concentration or ultimate BOD of the wastewater, S0, and we are assuming that the microorganism concentration present in the incoming wastewater is very negligible compared to the biomass concentration in the system. That is why we are not giving any X value here. And this is our aeration tank. The volume of aeration tank is VA and biomass concentration is X and substrate concentration or BOD concentration is SC. This aeration tank, we are assuming that it is a CSTR reactor, that means continuous flow stirred tank reactor. So what is happening? As soon as this S naught enters in the system, instantaneously the concentration will be changing to SC, whatever we have seen yesterday. So, that is what is happening here, and here the flow, it is 1 plus R into Q, and we will be having X and SC as substrate concentration and biomass concentration, and this is the secondary sedimentation tank, and here we will be having a sludge wastage of QW. So, if you want to see here, this is Q minus QW, because QW we will be wasting here, I forgot to put it here, and Q minus QW is the flow coming out, and SC is the substrate concentration or BOD, and XC is the biomass concentration, and a portion of the sludge will be recycled to the aeration tank. RQ is the flow rate, R is the recycling ratio, and SC is the BOD, because same SC will be coming, and biomass concentration is XR, because in the sedimentation tank the biomass will be getting concentrated and we are recycling the concentrated sludge. And always we use this X to represent MLSS. MLSS is nothing but mixed liquid suspended solid. So, if you have the that liquid, mixed liquid, so if you find out what is the suspended solid concentration in that liquid, that is known as MLSS. So, we assume that mainly that is biomass, or another term is there, that is MLVSS, mixed liquid volatile suspended solids, because in sometimes along with the wastewater, the solids along with organic solids, inorganic solids also will be there. So, what will happen, this MLSS will be containing organic and inorganic solids, but if you want to find out what is exactly the microbial concentration, then we have to go for MLVSS, and many studies have shown that the ratio of MLSS and MLVSS, it is like this. If you have the MLSS value, if you multiply by 0.8, that is MLVSS, or in the total MLSS around 80% is MLVSS, that means volatile suspended solids or fresh cells. Okay. And when we talk about the activated sludge process, another important term is hydraulic retention time. Okay. What is the amount of time the liquid is staying in the treatment system, that is the hydraulic retention time. So, that can be calculated by V by Q. V by Q is the hydraulic retention time, and another important term is biological sludge retention time, that is BSRT. BSRT is very, very important in any biological treatment system, nothing but biological sludge retention time. Okay. The definition is average time for which a unit of biomass remain in the system. If you allow a biomass to enter in the system or created in the system, generated in the system, how much time it is staying in the system, that is the biological sludge retention time. If the BSRT is more in the system, what does it mean? It means the biomass concentration in the system is more, because more and more biomass is getting generated and it is staying in the system for a longer time. So, that is the BSRT. So, it is represented by theta C, and in most of the design purposes, this is the most important criteria, theta C. What is the biological sludge retention time we are providing in a system? So, theta C, this is the mathematical expression of theta C. That means what is the time the biomass is staying in the system. So, how can we find out? That is nothing but the total biomass concentration divided by the biomass wasted from the system daily. Okay. Here ST is the total active biomass in the system, and dx by dtt is the total quantity of active biomass withdrawn from the system daily. So, we will be getting theta C in terms of days. Okay. And when dx by dt equal to 0, that means if we are not withdrawing any sludge from the system, that means whatever sludge is generated, entire sludge is staying inside the system. What does it mean? It means the theta C or biological sludge retention time of that system is infinity, because we know that theta C is nothing but total x divided by dx by dt. When the term dx by dt becomes 0, okay, naturally theta C will become infinity. The process modification of activated sludge, that is extended aeration system, is working on this principle. Okay, there will not be any sludge wastage or sludge recycling theoretically. So there the theta c will be infinity, and all the sludge whatever is generated in the system will be undergoing auto oxidation, or they will be in that endogenous respiration phase. So they will be getting auto oxidized. So if you want to find out the theta c of an activated sludge unit, we can find out like this, that is x into Va. This will give you the total biomass present in the system, that means in the aeration tank. So Va is the volume of the aeration tank and x is the biomass concentration per liter or per meter cube, divided by, and what is the biomass wasted from the system, that is nothing but Qw into x. Qw is the quantity wasted into what is the concentration of biomass in the wasted liquid, plus Q minus Qw into Xc. This is coming out along with the effluent from the secondary treatment plant. So it is nothing but 1 by mu, okay, because we have seen that what is this mu? Mu is the specific growth rate. Specific growth rate is nothing but the growth whatever is taking place per unit biomass, okay, or dx by dt by x. So this is nothing but x by dx by dt. So theta c is nothing but 1 by mu. So if you know the specific growth rate of the system, we can find out what is the theta c of the system. So if you want to change the theta c, because we have seen that this is the most important design parameter of activated sludge process or any biological process, how much time we can retain the sludge in the system. So if you want to change the theta c, because if you want to get different efficiencies or if you want to modify your system, okay, we have to change the design parameter theta c. So how can we change the theta c without affecting the volume of the tank, because once we design the tank, the volume will be fixed. So if you don't want to change the volume, okay, how can we change the theta c? So here from the equation itself we can, it is clear that if you change the Qw term, okay, because theta c is nothing but x into Va by Qw into x plus Q minus Qw into Xc. So if you change this Qw without changing either x or Va, we can change theta c. And another one is HRT, hydraulic retention time, that is V by Q, and theta c is the design parameter. This is very important. Now we can see how can we find out the theta c, or how can we, if we know what is the theta c, we can provide, and what is the influent BOD and effluent BOD required, okay, because we know that what is the treatment required, it should meet the discharge standard. So if in terms of BOD it is 30 milligrams per liter. So if you want to find out the volume of the tank, how can we go around? So what we'll do, we'll make the material balance equation with respect to biomass. So if you want to take the material balance, how can we do it? Net rate of change in the amount of biomass within the system is equal to rate of biomass production in the system minus rate at which biomass is leaving the system plus rate of biomass entering with the influent. So this is the biomass concentration. We will see, okay, this is the activated sludge process. We are taking the biomass concentration in the aeration tank. So we have to take, okay, what is the net rate of change of biomass concentration here, that is equal to what is the biomass generated here and what is the biomass leaving the system and what is the biomass entering in the system, okay. If you take all those three into account, we can get the mass balance. That is what we have done here. So if you want to write the mathematical expression for that one, it is like this: Va, this is the volume of the aeration tank, into dx by dt, change in microbial concentration, net change, that is equal to dx by dt gross into volume of the tank. That means whatever is generated in the system minus Qw into x plus Q minus Qw into Xc. This is the biomass whatever is leaving from the system. This is because of the sludge wastage, and this is going out because of going out through the effluent from the secondary sedimentation tank, and this 0 is coming because we are assuming that whatever biomass coming to the system is 0. We are assuming that the biomass concentration in the wastewater is very negligible compared to the biomass concentration in the aeration tank. And when we discussed about the biokinetic parameters, we have seen that dx by dt by x, that is the specific growth rate. So if you multiply that one with x, so we will be getting dx by dt growth, it is equal to yt into dx by dt growth minus kd into x. That means whatever is the yield coefficient into dx by dt minus this is the endogenous decay constant kd into whatever is the biomass present in shell. So if you substitute this one for dx by dt in this equation, the final equation will be becoming Va into dx by dt net is equal to yt into dx by dt g minus kd into x into volume, because volume is coming common, minus this term we can find out from the theta c equation. Theta c is nothing but x into Va divided by qw into x plus q minus qw into xc. So this term we can write it as x into Va by theta c. And we know that at steady state what will happen? The net biomass generation is equal to 0. Whatever is generated is leaving from the system, okay, or at steady state Va into dx by dt net equal to 0. So finally what will happen? Okay, this entire term will become 0 and we will be having 0 is equal to yt into dx by dt g minus k into kd into x into Va minus x into Va by theta c, or x is equal to theta c into yt into q into s naught minus sc by volume of the aeration tank into 1 plus kd theta c. So we can find out what is the biomass required for the particular treatment efficiency. S naught minus sc is the substrate removed from the system. That means this is the efficiency of treatment. For example, if you have a BOD of 300 initially and SC is 20 milligrams per liter, S naught minus SC is the one which is getting removed from the system, and KD we will be knowing and YT we will be knowing because these are the biokinetic parameters, and theta C is nothing but the 1 by mu. So X we can find out using this formula, and similarly we can develop an equation for substrate concentration. We know that mu is equal to 1 by theta C, which is equal to YTQ minus KD, and we know what is the definition for Q. Q is nothing but specific substrate utilization rate. What is the substrate utilized by unit biomass in the system? So Q is equal to ds by dt by x. ds by dt is the rate of change of substrate concentration in the system, or Q is equal to Q max SC by Ks plus SC. This is the maximum substrate utilization rate, and this is the half saturation constant. This all we have discussed in detail earlier, or we can write like this: ds by dt. What is the rate of change of substrate concentration in the system? That is equal to Q max into SC, whatever is the substrate concentration present in the system, by Ks plus SC into X. But in activated sludge process, what we assume is the substrate is limiting. That means as soon as it enters in the system, okay, S naught will be getting converted to SC because it is a CSTR reactor. So the biomass, the substrate or the food available for the biomass is always under limiting condition, or other way we can tell that compared to this Ks value, this SC value will be very negligible. SC is very less compared to Ks value. So how this equation will become? We can write like this: ds by dt equal to Q max by Ks. We have neglected that SC in the denominator into SC into X, or we can replace this Q max and Ks by a single constant small k, because Q max is a constant for a system and Ks is also a constant for a system. So two constants will give another constant, so that's why we are replacing that one with another constant k. So ds by dt is equal to k into SC into X, where k is Q max by Ks, or In other ways, we know that what is Q in an activated sludge process. Q is, what is the ds by dt? It is Q into S naught minus Sc, that is the total substrate concentration change in the system, and what is the total biomass concentration in the system? That is nothing but Va into X. So, Q is nothing but capital Q into S naught by Sc by Va into X, and we know 1 by theta c is equal to Ytq minus Kd. So, if you substitute this one here, we can get the value for Sc. We have to substitute this one here, then we will be getting Sc is equal to 1 plus Kd theta c by K into Yt into theta c. So, what does it mean? The substrate concentration as well as the biomass concentration in the system is depending upon the biokinetic constants of the microbial population present in the system. So, Sc is a function of Kd, this is the decay constant, and theta c is nothing but 1 by mu, and Yt is the yield coefficient, and K is nothing but Qmax by Ks. And another important thing in activated sludge process is recirculation ratio R, and how this recirculation ratio R is related with the theta c, that means biological sludge retention time. Okay. Again we can take the mass balance dx by dt equal to Rq into Xr, whatever is the biomass coming into the aeration tank, plus dx by dt into Va minus 1 plus R into Q into X. This is the biomass mass balance for the aeration tank alone. So, we can write like this, Rq into Xr plus, this time we are replacing by Ytq minus Kd multiplied by X into Va minus Q into 1 plus R into X. At steady state again this term will be becoming 0. So, we will be getting another relationship as 1 by theta c equal to Q by Va into 1 plus R minus R into Xr by X. Again we can see that if the theta c is fixed, the volume of the tank is fixed and the flow rate is fixed. Okay. The R is depending upon the concentration of the recycled sludge and the concentration of biomass in the aeration tank. So, here Xr is concentration of sludge in the recirculated sludge, Xr by X reflects the settling property of the sludge. That means how the sludge settling is taking place in the secondary sedimentation tank. That is very important for the proper functioning of an activated sludge process. If the secondary sedimentation tank of an activated sludge process is working perfectly fine, the entire process will be working perfectly alright. If it is the other way, the process will not be having the required efficiency. And how can we find out this Xr? Xr can be obtained from SVI. This term is also important. This is nothing but sludge volume index. Sludge volume index is nothing but the volume occupied by a unit weight of biomass. Okay. And usually SVI is represented in terms of milliliters per grams. That means if this much ml of biomass is there, what is the weight of that biomass, or what is the volume occupied by a unit mass of biomass, that is the SVI. How can we find out the SVI? Okay. So, before that one we will see how this SVI is important for activated sludge process. Small SVI is preferred. If SVI is less than 100, it is a good sludge. If it is 100, it is desirable. If it is greater than 150, it is bulking sludge, and if it is greater than 200, it is not at all desirable. So, how can we find out the SVI of the system? So, what we have to do is, okay, take a graduated cylinder and take 1 liter of the mixed liquid suspended solids in that one and allow it to settle for 30 minutes. Okay. After 30 minutes what will happen? We can find out the volume, because it is a graduated cylinder, we can find out what is the volume occupied by the sludge, and afterwards we find out what is the MLSS concentration in milligrams or grams per liter. From that one we can find out the sludge volume index. Okay. And sludge volume index is very important for the proper functioning of activated sludge process. So, now we will see what are the things we discussed today. So, we have seen that biological processes, okay, is suitable for either water or wastewater depending upon the pollutant nature, and Coming to the wastewater, what is happening? Most of the solids are highly biodegradable. So, biological process is the most desirable treatment process for secondary treatment of wastewater, especially domestic wastewater. Since the biological process is depending upon the microorganism for their efficiency, it is essential to provide sufficient nutrients, environmental conditions for the microbial growth, and depending upon the nutrient requirement, we can classify the microorganisms into different categories: autotrophs, heterotrophs, phototrophs, chemotrophs, etc. And depending upon the electron donor, we can classify the process into aerobic, anoxic or anaerobic. And we were discussing about activated sludge process, what is happening there. So, we are providing aeration. So, the dissolved form of organic matter is getting converted to more and more fresh cells in the colloidal form. Okay. And since more and more cells in the colloidal form is present, a solid liquid separation process is essential. So, secondary sedimentation tank is an essential part of activated sludge process. And how the sludge settling is taking place in the secondary sedimentation tank? It is with the help of some biopolymers which is excreted by the biomass themselves. Then we have seen that two parameters, one is hydraulic retention time and another one is biological sludge retention time. For any biological treatment system, the design parameter is theta c, not the HRT. Theta c is nothing but how much time the sludge is remaining in the system. And we have seen the mass balance of biomass and the substrate or B body in the entire system. And we have also discussed how can we find out the biomass concentration, the volume of the aeration tank, or the efficiency of the treatment using the biokinetic parameters of the microbial population present in the system. And another parameter we have discussed is sludge volume index. Okay, this shows the settling characteristics of the sludge, and this is also very important.